our very last speaker of 2025 at Nerd Night. Woo! He's going to be Patrick. We'll give him a round of applause and then I'll introduce him a little bit. Now, we need to make Patrick very welcome because he's actually the director of the institute where I work and the sponsor of Nerd Night. So, I think that's pretty cool. All right, so we'll welcome out... I was going to say the girls. We'll welcome out Patrick and he'll introduce his little guests halfway through his talk. All right, everyone, welcome Patrick.
Thank you. And just to start, we're very proud to be able to sponsor this evening. Thank you, everyone, for your support. And big round of applause for Ebony and Halo and all the team behind it. Excellent. All right, so I'm going to talk to you about my favourite things at the moment in the lab. This is an animal called Trichoplax, and we've sent it to space. I'm going to try to tell the story behind this and why this might be a good idea. So first, I'd like to acknowledge that we're on the unceded lands of the Wurundjeri people of the Kulin Nation. I'd like to welcome all Indigenous participants tonight and pay my respect to all elders, past and present. So this is Trichoplax. So Trichoplax is the simplest animal on Earth. Trichoplax stands for tricho, hairy, plax, plate. And you can see, extremely simple. It's about 800 million years old, one of the most ancient animals on the planet. And interestingly, and we've had a talk already on zebrafish, it has all the major gene families found in humans that regulate development and disease. And we use it as a medical model, and I'll explain why. It's also the fastest regenerating animal on the planet. If you do a little cut in it, within 2 minutes it heals. And so there's some very interesting things going on there. And to make it even more fun, you can pull it apart into single cells, and overnight you basically get many, many animals. So highly, highly regenerative. It's a part of a family of earlier animals. I think evolutionary biologists are still fighting on who's first or who was the first animal. We don't really care. This one is the simplest one. And that is the most important thing here. But you'll see there's sponges, jellyfish, hydras. All these are the early animals. And there's something to be learnt in all these. And all of them are highly regenerative too, which is interesting. And so it was discovered in 1883, and it was found by a German zoologist known as Franz Eilert Schulze in a seawater aquarium in Austria. I should say, in terms of size, it's the size of a grain of sand. So it's tiny, 1 or 2 millimeters. And it's extremely simple. It's got 3 layers, and this is at the bottom, you can see, 3 layers of cells. And it's got 9 different cell types. And although that might sound like a lot, humans have 200 to 400 different cell types. People are still arguing about it. It also has about 11,000 genes, probably about half of the number of genes found in humans, but still a large number for such a simple animal. And it has neurons, but no brains. So again, in a very interesting animal, how does this animal go around with just neurons and no brains? It feeds on algae, detritus and diatoms, I'll show you this. And also it replicates and divides in lots of strange ways. Binary fission, it just divides into 2. It buds. It also swarms. Bits of its body starts spouting out. And it also has sexual reproduction, except no one's seen it. But we know from the genes, analysing the genes, that they do reproduce somehow, somewhere, but never seen in the lab. So this is kind of the animals feeding. And as you'll see, we think of them as just guts wandering around to eat. They'll stop. The algae is fluorescent red. And as you can see, as it's digested and they sit on top of it, they put out their gut fluids and digestive enzymes onto their bacteria, onto the algae, and then will absorb it. So it's literally like an open gut that's wandering around, sits down, digests, moves on. And this is how they divide. And look at the scale, right? This is in minutes. This one will basically keep moving around. You'll see a little bit of a stretch, 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 stretch. And we've got 2. Okay, and they're clones of each other, identical. However, after a little while, you can actually transplant pieces of it to each other's, and they take the transplant, but after a while, you can't do that anymore, meaning that somehow these animals that don't have an immune system can recognize self from non-self. And there's something very interesting there also, because that could tell us about how we recognize self and non-self, which is not with the immune system. So again, some very interesting things about this animal. It's found all around the world. I would say it's probably one of the most adapted, well-adapted animal on the planet. It's been found in the water. It's usually found in the first couple of meters down on the beach. There's been a bias in terms of where they're found in warm waters, because that's where people go swimming. And so most of those studies have been kind of gathered from warm waters. However, if you look at the top of the map, you can find them in extremely cold waters, Scotland, and even colder waters, Melbourne. And this is where we find them. So we've been working with a dive club at the university, the La Trobe Dive Club, so thank you, Dive La Trobe, and we've worked obviously with Parks Victoria to actually collect these samples, and we've been going around the bay to see whether we could find Trichoplax there. And the way we do that is the dive club basically takes these little glass microscope slides, which you'll find here, they're just standard slides. We put them around the bay, and then 6 weeks later we take them back. And by that stage, a whole life ecosystem has actually grown on the slides. We get hydras, we get little worms, we get all sorts of algae, and we get Trichoplax. And in fact, if we keep these slides in the lab just with BL seawater, these last for about 6 months. So it's a very nice way to be able to collect these animals. And so why are we interested in this animal? What can we learn from Trichoplax? So I think a lot of people have been interested in these very first animals because one of the biggest questions in evolution is how do we go from single cells to multiple cells in animals? And the definition of an animal is actually not just an aggregation of cells, but an aggregation of cells that have different function and work together. And that's cooperation, and that's an animal. And the reason I got started with this, I'm a cancer researcher by training, is actually understand how single cells becoming multiple cells working together. You could imagine that if one cell decides not to play with everyone else, it outgrows the others, and that's the start of cancer. But none of these animals get cancer. So we're trying to understand how they do this and whether we might be able to apply that to human cancer. They also regenerate very quickly. So I think there's something about regenerative medicine that we can learn about these animals. And lastly, which I'll speak about, we know that these animals were around from the dawn of the ages, and gravity was around at that stage. And so we think that they might actually tell us about how gravity actually impacts on all the things on Earth in terms of biology. And that's why I want to tell you a little bit more moving forward. And so we got very excited about this because now we can play with space. So why would you actually want to go to space and do experiments in space? And this is kind of really our question that we keep on asking ourselves, why space and why should we actually go and work in there? So the first one that's pretty obvious, we have people up in space. We want to understand how long-term effects of space on human health for space exploration. To be honest, this is 5-10 people. It's not a huge number of people. We want to go to the moon, we want to go further past there, but we're not ready to be 6-9 months into space. But I think it's still an interesting question. What I'm passionate about is actually this other one. Space is a unique environment. We can use it like a lab where there's no gravity, and we can ask all these questions about why gravity might be important on Earth for Earth health and Earth applications. And the last one also has a unique environment. We can actually do things in space that we can't do on Earth. For example, again, from a biological point of view, stem cells seem to grow really well in space. And if we could understand why that is, we could actually take that knowledge and actually grow stem cells really well on Earth. So this is kind of the sort of interest in biotech. And so that question we're interested in is actually the role of gravity. And we think that gravity is really important for a lot of processes that is important for us. Cancer, regeneration, but I think just bodily functions. And as I said before, life evolved under the force of Earth's gravity. We know that we walk on two legs because gravity has actually sculpted our bodies that way. And I guess what we want to find out exactly how many of the different systems in our body are actually regulated by gravity. We know that when gravity is altered, cells, tissues, and organs show a lot of different effects. But probably the most striking is if you look at astronauts in space, they have a lot of issues. As with most elite athletes, they all underreport their problems. They want to be up in space. They can't hide bone loss, they can't hide muscle loss, but there's a lot of other problems there too, which tells us that we need gravity to be healthy. So what is it about gravity that makes us healthy? And just to put it in context, because we want to understand, for example, astronaut health, there's a lot of issues that they have to deal with. Distance, confinement, hostile enclosed environments, radiation, and gravity, which is important. And we know that gravity also, for astronauts, they've got impaired wound healing and regeneration. They can't actually heal very well in space. So we're, again, very interested in why gravity is important for this. So this started a project about 5 years ago between myself, Professor Bernd Shearwater, who's actually one of the godfathers of Trichoplax. He's basically been studying nearly by himself for half a century and knows everything that you might want to know about Trichoplax. And Dr. Jens Hauslager from the German Aerospace Center, who one day basically sent us an email saying, would you like to send Trichoplax in space? And that's how this project started. And we said yes. We had been working with Bernd about talking about cancer, and this is still a big interest of us, but today I'll talk to you about the space work. And Jens' interest was basically, he'd been working with small microorganisms, and this was the simplest animal. And so that made sense to try to utilize it to look at space. So the question we have is, how does gravity impact on the development and regeneration of this most simple animal on Earth? I've told you why we think it's important. And one of the reasons why it's actually a nice model to work in space is, one, it's tiny. I've shown you they can grow in lots of different temperatures in the waters in the Earth. So it means that it's actually quite resilient to temperature changes. And this is really important from a practical point of view when we actually prepare space experiments. And it grows in seawater with just minimal nutrients. So it's really easy to work with. So this is one of the first experiments that we did. So before we go to space, we want to actually understand on Earth how these animals might be reacting. And we can't simulate microgravity. So we simulate the loss of gravity, and the way we do that, we just bamboozle them a little bit. We move them around in a circle, they start losing track of where they're going, and that's kind of a way for us to simulate microgravity. So this is one of the contraptions we use. There's a camera here, and this spins. And the camera basically just records the animals moving around. And this is here, you can see the animals around here. It's spinning, and we can track the animals. And if we actually look at where the animals is, this is control, where basically there's no spinning. These animals actually like crawling up the slide of the tube. They actually go against the gravity vector. When we spin them, you can see they go random, basically meaning they've lost sense of up and down and gravity. So this is kind of some of the assays that we utilize. And we've been able to use that to look at the genes that change based on loss of gravity, and tells us a little bit about why these animals and how they're going to sense gravity. We know a little bit about how they sense gravities, and this is a kind of from my point of view, a very cool thing. So this is one of the crystal cells, one of the cells within the animals, the crystal cells. This is a side view of the animal. And it's got this very interesting little crystal of calcium carbonate, aragonite. So these animals actually make these little crystals, keep them in these cells. And what it does is that because it's weighed, it actually drags down, and the animal knows where up and down is. So that's how it uses it. In fact, it's exactly the same system we use to actually have our own balance and detect gravity. We have very similar crystals up in our inner ear, and as we move back and forth, that basically gives us weight, and we know up and down from this. So exactly the same thing, but 800 million years difference between the two systems. So we decided to actually look at what we'd call real microgravity. So before we go into deep space, we do very short-term experiments. And we use sounding rockets for this. So this is a collaboration with the German Space Agency. They've got a mobile rocket launching system called Moraba. And they've got a program called the Mayfirst Sounding Rocket Program that's been going on for about 15 years. And they go all around the world shooting rockets and shooting experiments into space. So we've been very lucky to be able to actually work with them in this. And more recently, they've been working in S-Range in Kiruna, which is in northern Sweden, near the Arctic Circle. And what it allows us to do is actually get about 5 to 7 minutes of microgravity. Doesn't sound like very long, but again, I've told you this is, for example, for wound healing, it only needs a couple of minutes. So we can actually devise experiments that can use this. From a space experiment, we call it late access. It means that we can actually go onto the rocket 2 hours before it takes off. We don't have to wait days. So this is very practical for us. And we can retrieve the samples a couple of hours, hopefully, or sometimes days later. So it means that we can actually analyze the samples when they come back. And the way it usually works is basically we go above the Kármán line. So the Kármán line is what people call space, 100 kilometres. And you would have seen Cape Perry come up to 100 k's for 1 minute. And then come back down. So they went into space. We're going all the way to about 250 to 300 kilometres into space, and the International Space Station is about 400, 450 kilometres. So it's close to the International Space Station. And then it comes back down. But what it does is basically gives us all this period with just microgravity. So this is where we do the experiments in Kiruna, near the Arctic Circle, because there's no one there, except for indigenous Sami tribes with their animals coming through and wandering around. And we have to basically stop the launches when those occur and let them basically settle. And then when they move on, we can have launches. And obviously, it's the land of the Northern Lights. This is my team here, and Melrose is one of the PhDs in the lab that's been doing the experiments. And Jeff is an engineer that helps us actually make these small labs that we send into space. And it's a beautiful place. It's in the tundra. There's nothing there, so I won't go into it, but it's also a logistic nightmare because we have to take thousands of reagents, thousands of things over into northern Sweden, and if anything's forgotten, we're stuffed. So we need to plan extremely well, and I've got an excellent team that's been able to do that. And so this is our first flight. So we have our space cuvettes, which are here, and you can see there's the Trichoplax in the centre. So they'll go into space with this. It's a pressured cuvette, so they don't feel the vacuum. And this was the mission control. So you can see we've got live feed of the Trichoplax being sent from the rocket. So this is the actual launch. A little bit of snow, probably about -10. And this is the first part of the rocket burning up, and then the second stage is going to come after this. So you'll see it there. And then they'll give it enough altitude to go through. About 500 kilos of experiments. There's about 15 teams, 25 experiments, a lot of experiments there, and a great team effort to get there. So this was this particular mission, Mayfield's 10. It got to 250 kilometres. We've got 6 minutes of real microgravity. And for us, it was about, can Trichoplax survive? We're talking about 9 Gs, sometimes 25 Gs, actually acceleration to get there. Trichoplax seemed to be very happy. So that was the great maiden flight. And seemed to be happy in real microgravity. So that was kind of the point of the experiment. Can we actually utilise this animal to actually get into space? And that was a resounding yes. And we had, again, live feeds plus all sorts of information about the rockets as we were doing this. And then we came back 2 years later. So now we want to understand what genes change during microgravity research. So this was Mayfield's 12. There's usually 1 launch or 2 every year. And this is my collaborator Jens, and again we're working with the University of Hanover. So here we're 3D printing everything. So these are just syringes. It's very simple tech. We basically have automated syringes that basically pump reagents onto the Trichoplax and can collect all the gene information that we need at the time that we need. So all automated. And this is what it looks like once it's actually inside the part of the rocket. We do a lot of testing, we have ground controls. There's a year of work to actually get to that stage. And this is the launch here. So again, lots of snow, again, -10 around October, November of the year. And again, a successful launch for this one. In this particular one, we were able to actually get the samples back 2 hours later. This is not always the case. Sometimes there's so much snow, the helicopters can't go and get them. This year, we also had a launch a few weeks ago I came back from, and we had to wait 72 hours. There was a snowstorm, everything's frozen when you get it back, so you have to plan for these things. A few years ago, there was so much snow, they had to wait till the spring to get the samples back because the beacons on the rocket basically lost battery, and then it was impossible to find afterwards. So Trichoscope was the very first one, a microscope looking at Trichoplax. Graviplax, we're scientists, so we have to do things at least 3 times in repeats, and this becomes quite expensive. One rocket campaign costs about 6 million euros, $12 million, and we've been very grateful to the German Space Agency to actually be able to ride share with them. So we now got lots of genes to look at. We're testing those to see how important they are for gravity. We want to look at the human genes because we know that many of the human genes, maybe the genes in Trichoplax match human genes. And our next mission is really about looking at this process of regeneration that we know is super fast in Trichoplax. We can do simulated microgravity. We've got 6-7 minutes on the rocket. So the idea is to have probably just a bed of needles to be able to puncture the Trichoplax and actually look at healing in that particular case. And that's the next set of experiments we're getting ready for. So what's next for Trichoplax? I've told you about all the big questions. We're still very interested in cancer and regeneration. But I think gravity, understanding the basic way that gravity impacts on health is, I think, a really good question to ask. We're doing some work at La Trobe on sleep, and that might sound odd, but there's a theory, which is more than a theory, about sleep being for repairing the DNA in your brain as you sleep, right? Lots of free radicals and toxins which are produced when you're thinking, when you're alive, when you sleep, all this is repaired. Trichoplax doesn't have a brain. So the idea is, do you need a brain to sleep? Can you sleep without a brain? Is it even more fundamental? And what does sleep mean for these sort of simple animals? I've told you about self from non-self. And then there's a lot of other things we can do, which we're very keen on. And part of the reason for working with Trichoplax, such a cheap organism, there's a lot of fundamental ecological questions that have been asked about utilising to look at the health of the oceans, for example. And so we've been very keen to actually have more and more people involved in researching this. And so I wanted to kind of introduce you to kind of citizen science and our youngest nerd-niters, which will close off this talk. So I'd like you to welcome... Would you like to introduce yourself?
Yeah. Hello, I'm Lucia.
And I'm Jonah.
And we're from... and we're from St. Monica's Primary School from Footscray.
And today we're going to present our research on Trichoplax. Our aims were to culture Trichoplax at home and to test which conditions Trichoplax grow best in.
Our hypothesis is that Trichoplax adherents will grow best in light conditions because the samples were obtained in shallow water between 1 and 3 meters deep, meaning that the rocks that they live on would have been exposed to the sunlight.
These are the materials used to culture and monitor Trichoplax at home.
Collecting samples from Williamstown Beach. We went to Williamstown Beach to collect rocks for our experiment because Trichoplax grows best on rocks.
The rocks hypothesized to contain Trichoplax were collected at 1 to 3 meters deep using standard snorkeling equipment.
And we put the rocks into mason jars filled with seawater.
A microscope slide was added to the jars because Trichoplax loves growing on glass. The Trichoplax was cultured for 1 month.
Monitoring Trichoplax culture environment. We monitored temperature, salinity, and pH in the jars for a month. This is important because Trichoplax likes to grow at 19 to 21 degrees Celsius, 3.5% salinity, and a pH of 7, which is neutral.
After a month of culturing, images of the cultured Trichoplax were captured on a Leica inverted microscope times 100 magnification.
This is the Trichoplax that was growing in the mason jar in the light.
Indentations in the Trichoplax signal that it is about to divide.
Thousands of Trichoplax have since been cultured from this single mason jar.
Genotyping is being performed by LIMS to establish the genotype of Williamstown sample. Initial results indicate it may be either a H5 genotype, previously not discovered in Victorian waters, or possibly a new genotype altogether.
Conclusion. Our results support the hypothesis that Trichoplax is best grown in light conditions, and our experiments prove that Trichoplax can be cultured at home using household items.
Thank you to Professor Patrick Humbert and Dr. Samantha Melrose and lab for supporting this collaborative study.
Fantastic stuff, guys. So, just a couple of questions. If you were going to do this again, how would you do it differently?
That's a good question, Pat.
We haven't rehearsed.
We would culture for longer if we had more time.
Well, Patrick, we would collect more. Wait, what did he say? Collect more samples. Oh, we would have collected more samples. Yeah.
And what was your favourite part of the project?
Probably snorkeling.
Oh, same Z's.
All right, please, a round of applause for our young people. Thank you.
All right, do I have any questions for Patrick and the squad?
Come on, guys.
All right, right here.
Awesome, thank you so much. The first question that I have would be for Patrick is, have they been able to be found in the fossil record? And then the second question is for the girls. Can you tell us a little bit more about like the salinity, the saltiness of how that affected the.
Okay, so the first question was for Patrick, and have they been found in the fossil record?
Yeah, so I think a very good question, and I think that's a million-dollar question. They're soft-bodied, and so they disappear very quickly. However, I think we should still try to think about whether we can actually find them maybe in the gut of a predator. Unfortunately, there doesn't seem to be a predator for these animals. One of the interesting things that I haven't shown you, but these nerve cells that they have actually secrete neuropeptides, and these are a bit like venom. And if you watch them, you can see little creatures going to try to attack them, and as soon as they touch them, they just run away. So those nerve cells actually seem to be protecting them in some ways, but no fossil records would be a great thing to see.
And your second question was for the girls?
Yeah, I was just wondering if you could tell us
Boy and girl.
Sorry, boy and girl.
That's okay.
I was wondering if you guys could tell us a little bit more about how the saltiness, how you saw that.
If it's not the right conditions, the Trichoplax can find it hard to grow, so we need to keep it at stable conditions.
Okay, another question at the front here.
You might have mentioned it, I might have missed it, and it goes a bit on the work that you guys done. Are we talking about a single species?
Yeah, so I didn't mention it. So there's about 20 species. They all look exactly the same. As opposed to, say, some of the other basal animals that I showed you, where there's tens of thousands of species with different body plans and all sorts of magical variations. These ones look like blobs. 19 out of the 20, and the 20th one looks like a blob with more blobs.
Are they geographically distributed?
No, they're everywhere. So, two interesting facts. One of them is they stick on ships, and so they just go around the world. And there's another interesting paper looking at what's called eDNA, environmental DNA, where basically you just scoop the top of the water around the seas, and this was a study in Japan. And if you do that, the most common DNA for animal DNA is Trichoplax. And so although we think of them sitting on rocks, they're actually in all the water columns in the oceans and on the top of the surface. So it may be that we're not thinking of them properly as crawlers, they might just be swimmers. And we can see them swim too, but that's not usually how we think about it. But they're everywhere, so there's no particular species for anywhere.
Yes, one right here.
Hi, I just had a question for Lucia and Jonah. I was just wondering, like, what intrigued you about learning about Trichoplax, and also, like, how did you know to contact Patrick without learning about it?
So this one was for Lucia and Jonah. What intrigued you about Trichoplax, and how did you go about contacting Patrick here?
So we saw like some website about also Trichoplax, and like we learned a lot about it lately on websites, and it looked like it was from Patrick's website, yeah, and it looked very like fascinating, and we wanted to learn about it, yeah.
And we wanted to help get research for Patrick, so that's why we contacted him.
Do you have anything to add, Patrick?
Well, it was, it's been I must say, it's been a fantastic collaboration. I talked about, I guess, more the kind of citizen science, and we need to know more about this animal. It's everywhere. We know that in healthy waters it thrives, and I think our collaboration has shown that we can actually do this in Melbourne. It's cheap. We can find new information about new species. The species we've found, we're still trying to analyse. There's something strange about it, meaning that the DNA is closest to the H5 species, but it's not H5, it's something different. So we're trying to understand that. And it's been wonderful to work together. Thank you for coming tonight.
Okay, wow, a lot of hands up. Firstly, up here.
Thanks, Critical. Any plans for the ISS with Trichoplax? And for the little one, when do you start the PhD?
So, Patrick, any plans for the ISS with Trichoplax?
Yeah, so as everyone will know, the ISS is going to be decommissioned in 3 years' time. There's a huge queue of experiments waiting to go on there. We're trying to see what else is happening. The Chinese space station is up there and fully operational. There's a lot of worry that we don't want just that space station to be operating by itself. Elon Musk and all the other tech bros are kind of sending their own versions of space hotels up there, but it'll probably take a few years for us to be able to access that. With the German Space Agency, we've got plans to actually try to get something in the ISS. They've got a very nice microscope that does multiple colours that we might be able to use up there. We've applied for it. We not failed, but we weren't successful in our application to access last year. But hopefully we can actually get there in the next couple of years. And I would say a lot of the work that we're doing, the ground-based work, the sounding rockets, although it can be limited in the short amount of time, is all preparation for these long-term space missions.
And the second question for you guys, and the question was, when do you start your PhDs? But do you know what you want to do at all? Keep doing science? Keep doing citizen science?
Probably science.
Yep.
Yeah, science.
All right, this hand at the back here.
So you said that Trichoplax could be used for monitoring water health. What's better, Trichoplax or zebrafish water?
I missed the question, but what?
We're about to wrap.
Patrick's my boss, I should say, so I'll just be very respectful.
No, I guess the interesting thing here, I should say, is that you'll see that both of us are using marine organisms to actually learn about human health. And this is really about utilising lots of these Each model has limitations and advantages. For us, they're simple, cheap. We can do this in lots of different ways. We can send them in space. Zebrafish have been sent into space.
We should do that, actually. I think that's a really good idea.
So this is actually
We should chat.
So we're collaborating, not working against each other.
Which is better for monitoring water health?
Oh, which is better for monitoring water health? Zebrafish.
I think Jason Statham might be better for it.
Jason Statham's been to space.
All right. Okay. Okay. Ebony's saying only one more. We have to wrap up. Who wants it the worst? Everyone's pointing towards this person at the very back, so.
So you said that they don't have an immune system or brains. Do they show any evidence of memory in any kind?
Do they have memory?
I think it's a very good question. Some labs are basically arguing that, and this, I would say physicists, I hope there's no physicists in the room, but physicists are basically saying that because of how simple they are, we should be able to actually compute them and be able to predict everything that they do, right? So I think memory is an interesting one to actually think about, whether they could remember where the food source is, for example, because in some ways you think they're so simple, they're just literally just reactive to what's going on. So a very good question whether there actually is some sort of form of memory. Thank you. That's going to be our next NHMRC grant. Seb?
All right, put your hands together for Patrick and the squad. All right. Unfortunately, that wraps up our final talk of 2025. What a year. 6 sold-out shows. Holy moly. We do a little spiel at the end of every show, and I guess this is also the end of year spiel now. So if you didn't know, we have a YouTube that is fully functional now, and we've uploaded 72 previous new Nerd Night talks on it. Yeah, it was a big effort. One of our volunteers, Matt, actually did it, so he's
Yay,
not
Matt.
here, but yay, Matt. So here's the QR code. If you want, background listening, anything to binge, 72 Nerd Nights right there for you to look at. And yeah, we're 100% volunteer powered. So at the end of every one of our nights, we like to thank our volleys. We was correct. We'll start with us.
We'll thank ourselves.
We'll thank ourselves. All right, let's go down the line. Nick, our DJ.
Who's not here, but up here.
He's up here somewhere. Nick. Michael, our videographer, making that YouTube come true. Moxie on photography, somewhere. Luke on sound and lights back here. David, who made our sick artwork you've been seeing all night.
He lives overseas, so he couldn't make it tonight.
He couldn't make it. Phil, who made our theme song. Who is
He's
up
at the back.
here somewhere. And Matt, who made our YouTube happen.
Yeah.
Howler for hosting us.
Woo! We'll be back here again next year.
Next year.
Keep your ears peeled for some dates.
Keep your ears peeled.
Yeah, it's the end of the night.
Yeah. Where are our cells? Where are my cells, sorry. QR code there if you want to look at your Christmas shopping for any nerds, go ahead. Latrobe for sponsoring us.
Limbs, specifically.
Oh, Limbs specifically, not Latrobe, Limbs.
Nah,
sorry.
Limbs.
And I'd like to welcome them back onto the stage. Tonight's speakers, we have so many.
We'll have all of the guests
All
as
of
well.
them.
Everyone, come.
All right.
We actually only have 3 mugs. So these will just go to the people that were named on our website. And we might have to send some things in the mail for the rest.
Magical, thank you.
There you go. And we have some stickers.
All right, put your hands together for these guys.
Everyone take a bow. You don't have to tell these two twice.
You too. Okay, lastly, we need you. We're filling up our 2026 calendar.
We might just part the ways here so people can...
Sorry. Sorry, beautiful people. We need you. We're filling up our 2026 calendar. If you are a nerdy person and you have something nerdy to talk about, or if you have a nerdy friend, please get in contact on like literally anything. We're on like every platform, I think.
Yeah.
Yeah.
We are.
We are. Email us, everything. Get in contact, please. And thank you for attending and making nights like this happen.
Woo!